so probably if you just have a little bit left on an old one, you should restart a new one for tomorrow. To try to cut down on the inoculating time a little bit, we're going to work in groups of 3 for inoculating known organisms, so that'll just be 6 groups of 3, so I'll pair you off to do that, and then that way we can divide up the known organisms a little bit. Okay, there's really only 3 more labs, 3 more regular labs. And they're fairly extensive, they've got a fair amount of information associated with them, and that will be the bulk of the information for the second lab exam. So you won't have quite so many labs to deal with, but there'll be more information in those particular labs. So be aware of that. We'll talk more about the second lab exam coming up in a few weeks. So what is this kind of esoteric, somewhat, philosophical sounding statement refer to? You're not really just you. You've got a lot of microbes associated with your body. What's the ratio approximately? How much of us is there, eukaryote, versus how much prokaryotic is there? About 10 times prokaryotic compared to eukaryotic. Up until just recently, people have not really understood the significance of the microorganisms that are associated with our body. With one exception, and that is the disease-causing microorganisms. We've known about the germ theory of disease for, over 150 years, which is a relatively short time in the span of biological science, but it's been around for a while. And medicine has been particularly concentrating on that idea of bacteria that cause disease, pathogens, are really the only bacteria we should be concerned about in regard to human health. And as it turns out, that's really a short-sighted point of view. And that what we're finding out now is this entire community of human cells and bacterial cells associated with our body, which we call the microbiome, is really important in defining a lot of attributes of us. That was one of the articles that I assigned early on. There was an article that talked about some of the things that your microbiome bacteria did, and it influenced things including possibly behavior traits, how you digest what you eat, and whether you gain weight from it, for example. There are just all kinds of connections that we never appreciated before about our microbiome and what a role it plays in our functioning and development. So in 2007, a pretty ambitious project was launched called the Human Microbiome Project, and it was kind of equivalent in some respects to the Human Genome Project. The Human Genome Project, remember, had the goal of sequencing the entire genome, and we did that. And actually that was really kind of an interesting story, because the Human Genome Project is probably, at least in the last probably 2 or 3 decades, a government-funded project. Does anyone know who funded the Human Genome Project? Department of Energy. Okay? A government-funded project that came in under deadline and under budget. When does that ever happen again? In part, that was the result of a partnership between government and private institution. And it was really driven a lot by advances in computing and the tremendous wave of computing power that developed over the time of the Human Genome Project. The Human Microbiome Project hasn't really developed yet that public aspect to it. So it's really more government funded through university research. But the goal is to try to identify the microorganisms, the communities that occupy various regions of the body. And for now, they're concentrating on the oral cavity. Okay, you did a little quick swab of your oral cavity just to see if you had Staph aureus or some strep species. But the microbiome project is much more ambitious than that. The skin, the nose, we did the nose too, the digestive tract, and the reproductive tract. Some of the particular outcomes of this are driven by a new strategy. The microbiome project is depending upon more than just trying to culture microorganisms the way we did. Take swabs from these things and then see what grows on plates. What's wrong with that strategy, and why might that strategy be limiting?
Certain bacteria might not grow on those plates.
Yeah, certain bacteria might not grow on those plates, okay? We talked about just the concept of a fastidious bacterium, for example. The last exam, do you remember what those are? It's a bacteria. They require some kind of supplement to the normal just TSA agar. Okay? So if you're really going to survey the oral cavity, for example, you would have to create all of this different media that might pick up the individual nutritional needs of every different possible species that was there. Instead, they're using a strategy called metagenomics, where they're screening for unique ribosomal RNA sequences. And the idea is, if you find a novel ribosomal RNA sequence, that is indicative of a unique species. So this really is going beyond just trying to identify what you can culture. It's really trying to identify what might be there, what is there, that we can culture, but that still has a role to play in our body. So it's a very ambitious type of project. And new information is emerging constantly that is broadening our horizon about the role that our microbiome plays in us as organisms. And this is pretty new stuff. Well, microorganisms don't exist alone in nature. Okay? They're associated with our body. They're found in complex community on non-living surfaces. Two major types of relationships that microorganisms can be in: symbiotic relationships and non-symbiotic relationships. We're really going to concentrate on the symbiotic relationships. What does the word symbiosis mean? You know what sym or syn means? Together, okay? And bio refers to life for living. Okay, so symbiosis means literally living together. Organisms live in close relationships with one another, and that relationship is usually nutritionally based. So if we take the most simple example of two organisms living together, and let's say it's a human host and a prokaryotic cell, and we represent the outcome of that relationship by one of three symbols. Plus indicating benefit, minus indicating harm, and zero indicating neutrality, really no effect. Okay? So, what's mutualism? Well, mutualism would be designated how symbolically? Plus plus, right? Both organisms benefit from the relationship. Both are interdependent upon one another. We talked about a non-human microbial symbiotic mutualistic relationship when we talked about the nitrogen cycle, and specifically the symbiotic relationship between legumes and the bacteria that live in the nodules. The bacteria get carbohydrate from photosynthesis. The plants get fixed nitrogen from nitrogen fixation. Well, do we have mutualistic relationships? Absolutely. Some of the earliest ones that were identified were with our enteric bacteria, our gut bacteria, and us. Gut bacteria can produce some useful substances as a result of their metabolism, including some vitamins, like vitamin K. We're now starting to appreciate more the role that the gut bacteria plays in terms of suppressing other potential pathogens. And so that beneficial organisms, the organisms that live in our gut, still get a supply of food, okay? But by their growth, they then inhibit other organisms that might cause us problems. So a number of mutualistic relationships are found between humans and bacteria. Getting back to that idea that not all bacteria are bad. Okay? Commensalism would be symbolized as a plus zero. One partner benefits, the other not affected. When we did skin swabs, one of the bacteria that many of you found was Staphylococcus epidermidis. Okay? Those bacteria are adapted to living on your highly salty skin. They can derive nutrients from your dead, sloughed skin cells, so they benefit. Does it really matter that you have that Staphylococcus epidermidis growing on your skin? No. That's a very commensalistic relationship. The relationship that we're going to concentrate on pretty almost exclusively for the rest of the semester is parasitism. Parasitism would be symbolized as a plus-minus relationship. The host is going to be potentially negatively affected. The parasite will gain some benefits. What benefits? Usually nutrients. Okay? In a parasitic relationship, there is usually a competition between the host and the parasite for nutrients in the body. Depends upon how that teeter-totter is balanced in terms of what type of parasitism we have. Sometimes the teeter-totter is pretty evenly balanced, and the parasite does obtain some nutrients from us, but not enough that really harms us. Okay, they're a minor kind of annoyance, really. But you do have organisms growing in your tissues. They're there. Technically, we'll see in a minute that that indicates you have a state of infection. Okay? Infection refers to growth of microorganisms in the tissues of the body. That's infection. But if the parasite doesn't really gain the upper hand, and it just kind of uses some of your resources, but not enough that it really makes a difference, then we refer to that as a benign parasite. And yes, we do technically have an infection, but it doesn't really produce any symptoms. So we refer to that type of infection as a subclinical infection. And we're all probably carrying around parasites that are benign, and we're in a state of subclinical infection. Now, particularly if your immune response is pretty good, then you probably won't have any symptoms at all. However, if your immunity starts to lower, then the balance can shift. Okay? And the parasite can start outstripping the defense systems of your body, and now the teeter-totter balances more in the favor of the parasite. Okay? If that happens and the parasite gains the upper hand, then we develop a clinical disease as opposed to a subclinical. And that typically is associated with particular symptoms. And one of the things we're going to talk about is, why do those symptoms result? What is it that the parasite's doing that actually causes the symptoms to appear? Sometimes the competition becomes so severe and the parasite gets such an upper hand that ultimately the condition becomes lethal. If that's the case, the parasite is oftentimes referred to as an inept parasite. Why that terminology? Why would we consider a parasite to be inept? Killed the toast. Yeah, it's got to find a new host then, or it dies if the host dies. Okay? So sometimes pathogens that are very strong or very virulent, virulence is really a measure of how bad the pathogen is or how severe the disease symptoms are. Sometimes very virulent parasites really burn themselves out by killing their host too quickly before there's a chance for the parasite to jump to another host. Anybody think of a good example of a highly virulent parasite that has kind of a self-limiting infection, after a while kind of burns itself out? Malaria, possibly, although that you can survive with malaria for quite a while. Not Ebola. Okay? Ebola outbreaks tend to be very pop up, a lot of people die, and then they die back down again because the parasites are so virulent that they kill the host before there's time for really much transfer to other organisms. Well, if you look at the human body, you're going to find that all of these particular relationships are present in the human body. One of the things that's interesting about the human body is it's not a uniform environment. Think about the human body for a second. It's a wonderful incubator. It is thermostatically controlled to maintain a constant internal body temperature of around 37 degrees Celsius. It has a fairly constant set of resources coming in through our digestive system. We have elaborate mechanisms involved to maintain osmotic concentration of the body fluids at a homeostatic amount. The pH is regulated. Okay? All of those factors that we said influence microbial growth are really regulated very highly in the human body. But the human body is not one consistent type of habitat. It consists of a bunch of really microhabitats, each of which have unique conditions of temperature, pH, etc. And different microorganisms are adapted to different habitats. So it's not surprising that when we expect to find different populations of organisms in different parts of the body. And that the microbiome is not going to be the same in the oral tract, or the reproductive tract, or the skin, or the oral cavity, or the nose. There are going to be differences. That's to be expected. Well, these relationships between us and our microbiome all begin with a common denominator, and that is we have to come in contact with the microorganisms. Shut down a little bit for a second. So what happens when you come in contact with microorganisms? Well, you can become colonized with more or less a permanent flora. And we refer to this as your normal flora. These are microorganisms you acquire at birth. Now, does the nature of your normal flora change? Yes, it can. We'll talk about some of the factors that can influence your normal flora. You may know why the word flora. What does flora typically refer to? Plants, okay? Flora and fauna, plants and animals. So why do we refer to our microbiome as the flora? It really goes back to that old Linnaean system of classification. And in the Linnaean system of classification, everything was initially classified as either a plant or an animal. And the microorganisms were put in with the plants. So we retain that kind of terminology. Okay? Sometimes, however, the microorganisms cross the lines of defense and enter what are normally sterile tissues, like the blood, or the cerebrospinal fluid, or the internal organs. And if that happens, by definition, we have an infection. Now, maybe it's just a subclinical infection. Maybe it's a pretty benign parasite. At which point in time, you become a carrier. The microorganisms become established in the tissues, but there's no real disease state. Some of you have found that you are carriers of Staphylococcus aureus. It's not making you sick. But you're going to have to be careful, particularly if that's part of your normal flora, as opposed to your transient flora. We'll talk about those first too. So if you're a normal flora person for Staph aureus and you're in the medical profession, you really need to know that. You need to be very careful about hygiene, for example, and not transferring these bacteria, especially to immunocompromised patients. If you do have an infection that causes disease symptoms, hopefully your defense systems will hold the pathogen in check and eventually wipe out the infection and repair the damage. Sometimes you wipe out the infection, but damage persists. And you actually have complications after the infection has been resolved. Those complications are called sequelae. What is a potential sequelae of a polio infection? Yeah, paralysis. Okay. Franklin Delano Roosevelt, our president, had polio. He had recovered from the polio infection, but it left him paralyzed. Sometimes, as a result of the infection, there are various outcomes as a result of the disruption of the tissues that comes with the infection. And when that happens, we can have either illness or mortality. So let's take a look at just a second for this kind of outcome right here. You get colonized with some type of bacteria, and they take up more or less permanent residence with your body and become part of your normal flora. Okay? So here are some sites of the body that normally harbor resident flora that you pick up pretty much immediately after birth and actually during the birthing process. One of the things that we've observed over the years is that there are some differences in outcomes of vaginally delivered children and those who are delivered by cesarean. And one of the things that happens in vaginal births is the baby, as it's going through the birth canal, starts to pick up a normal flora from the mother that you don't get by the cesarean. And they've noticed some differences in development for children in those two populations. So the skin, upper respiratory tract, GI tract, outer opening of the urethra, genitalia, vagina, external urine canal, external eyes, okay? All of those can pick up a normal flora. What we're finding out is that normal flora can be pretty important to us and our function. And some of those insights have come from research done on azenic animals. Azenic means germ-free. This is just a shot of some typical normal flora bacteria that might be associated with your epithelial tissue. Azenic animals are produced if the mother delivers by cesarean in a sterile environment. Because while the baby's in the womb, the mother's immune system protects the baby, and there should not be infectious agents exposed to that child when it's in the womb. So if you deliver it in a sterile environment, you can have an azenic animal. They've done some comparisons between azenic and non-azenic animals and looked at what the effect of the germ-free state might be. They've worked with dogs, cattle, rats, etc. And over here you can see some significance that they have kind of concluded about the importance of that normal flora in organismal function and development. So microbes are needed for normal intestinal development. Microbes are a significant nutritional source of vitamins. They're needed to stimulate certain host defenses. Has anybody ever heard of the hygiene hypothesis? This is a hypothesis that suggests that some of the maladies that we're suffering right now. When I was growing up, for example, I knew absolutely no one with an inhaler. And the rates of asthma were just pretty much non-existent. Now, probably all of you have encountered people, if you don't do yourself, having an inhaler. Okay? Why is asthma increased? There are a number of kind of autoimmune diseases and other childhood diseases that are really seem to be much more common right now. And what some people attribute this to is we become too clean as a society and too obsessed by cleanliness. And everybody's got to be using antibacterial soap all the time. And everybody's obsessed by running around with the waterless hand cleaners and things like that. Kids don't get to play out in the dirt a lot anymore. Houses have filtration systems that try to keep bacteria and mold spores out. The conclusion from the hygiene hypothesis is that exposure to those microorganisms is necessary to stimulate your immune response, to have a normal, robust immune response. Because many of these conditions are being traced back to suppressed and malfunctioning immunity, including allergies. A lot more allergies right now, too. So this certainly hasn't been demonstrated without question, but there's a lot of evidence in support of this concept of the hygiene hypothesis. So if you have kids, let them go out and get dirt. Don't be too obsessed as a parent by constant cleanliness. Normal bacteria residents are antagonistic against pathogens. Break down indigestible carbohydrates, increase fat storage. There's just a long list of things that they've noticed about kind of deficiencies in azenic animals, leading to the conclusion that these microorganisms play an important part in your body. This particular slide gives you a fairly good summary of some of the parts of your body and some of the normal flora that we find in these particular locations. And this is not something that I would ever want you to memorize, but it's something that's kind of interesting to look at and kind of see what typically is in various parts of your body and what the consequences are of having normal flora in that particular location in your body. Now, importantly, there are regions of your body that should be relatively germ-free, if not sterile. And if these particular regions become infected, then we've got a problem. Then we've got a disease state with symptoms, and sometimes we've got a lethal condition involved. So we're going to talk about some of these later on. What happens when some of these parts of the body become infected? Remember what we call a blood infection? It's called septicemia. Septicemia is very serious. Okay, once you get microorganisms in the blood, it can be really difficult to treat them. Well, in addition to your normal flora, we all have some organisms that come and visit us and stay for a while, but eventually leave. They don't really leave. They die. They're not replaced. And those are called the transient flora. And you pick these up from other people. Typically, your transient flora don't cause disease if your immune system is in a good state. But if you have a lower state of immunity, you're under stress, maybe you're ill from another disease, then those transient flora can cause problems. Okay? A lot of people are carrying around as part of their transient flora Streptococcus pneumoniae, which is a bacteria that can cause bacterial pneumonia. What is pneumonia? What makes something pneumonia?
Not a technical, but I know that it's just fluid that gets built up in your lungs.
Absolutely. It's accumulation of fluid in your lungs, and that is a result of inflammation. Okay? Well, why aren't those transient streptococcus causing enough inflammation to cause fluid buildup in your lungs if you're healthy? Because they're kept in check by your immune system or by other microorganisms. But let's say you have the flu or a really bad cold, and your immunity suffers as a result of that. It's kind of fighting off another infection. Then those Streptococcus pneumoniae can start growing more out of control and can cause an inflammation. And so you can develop a secondary pneumonia as a result of immunity being lowered by a primary infection. And so that's why they particularly urge elderly people to get pneumonia shots, okay? Because if you do have flu then, you don't want to have the complication then of a secondary pneumonia resulting from that. Your chances of getting pneumonia certainly increase as you become more elderly because naturally your immune system declines with age. Your immunity gets lower and lower the older you get. Well, what we're really going to be most interested in for our discussions is the process of infection. So remember that infection refers to the growth and multiplication of microorganisms in the body tissues. There is some terminology. I've seen a slide like this for the last two topics we've done that talks about some terms associated with infectious disease. And I'm going to ask you to look at this particular slide particularly so you know what some of the terms are as we encounter them. So there's a fairly long list: acute, chronic, subacute, asymptomatic, latent, communicable, contagious, etc. A couple of these I've mentioned already, but you need to become familiar with these particular terms if you're going to be able to speak about infection intelligently. If we actually develop a disease state, then typically there will be some symptoms associated with that particular disease state. One of the questions we're going to dig into a little bit is, why do you develop symptoms? Well, there's a couple of possibilities. The growth of microorganisms in those tissues may damage them, may cause dysfunction, cell death. This is typically a result of various biochemicals that the pathogen produces These biochemicals are referred to as virulence factors. Remember, virulence is a measure of how nasty a disease is, basically. So as they grow, these microorganisms produce these substances, these virulence factors. The production of these is oftentimes an advantage for the microorganism, because many of these allow the microorganism to spread throughout the body and to increase its foothold. But typically, the accumulation of these biochemical substances and the damage that they cause results in particular disease symptoms. So as we kind of go through some of the virulence factors that pathogens possess, we're going to talk about some specific diseases and see how those factors result in particular disease symptoms. Sometimes, if you're really good, and it's an art, really, a good physician can fairly accurately identify the pathogen that's causing the disease by the symptoms that it produces. Now, if the symptoms are fairly general, then it becomes harder. But if the symptoms produce some particular characteristic outcomes, then it becomes a little bit easier. Unfortunately, many pathogens initially produce kind of the same generalized symptoms. What are some common generalized symptoms that many pathogens might produce? Fever? Coughing? Breathing problems? Nausea? Diarrhea? Okay. Kind of generalized things. And sometimes, by just looking at those general systems, it's pretty tough to tell. And sometimes it's even tough to tell the difference between colds and flu, which are caused by different kinds of virus. What's really important is taking a good patient history in terms of trying to nail down what a particular pathogen might be. And one of the things you'll learn if you go to medical school is that a patient history is not all just about the vitals. But there's a lot of things that you need to get in a good patient history that go beyond just those kind of baseline blood pressure, pulse rate, etc. I mean, those can provide you some interesting information. Temperature certainly can. But a good history really is more comprehensive than that. What are some other things that could potentially influence your outcome of diagnosis? Well, do you know anything about the person's profession? Or hobbies? Or where they've traveled? Okay? If you know that a person's traveled to a particular part of the world, then some possibilities for different types of disease agents all of a sudden become possible. If they don't know that Jake went to Africa this summer and he comes back sick, they're not going to be looking for some of those diseases that are more endemic to that part of the world. Why professions? Because you might be exposed to different things in your profession. Your hobbies might put you in exposure to certain things. There are people who like to work with leather, for example. And evidently, cowhide particularly is not uncommonly associated with anthrax spores. And so someone who likes to work with leather has a higher risk of anthrax. Anthrax is probably not something that would be on your radar as a physician unless you had that piece of information. And this is where it's getting kind of unfortunate that the way medicine is having to be practiced right now is that a physician typically has about 20 minutes with the patient. If you talk to a lot of older doctors, they say that should be at least double. That an office visit for a physical should be about 40, 45 minutes so you can get a more accurate, better patient history. So you've got time to talk to your patients and see what's going on with their life. What other kinds of things should you know about what they do? That's really important in that diagnosis process. If you're infected, there are some factors that influence how likely it is you're going to get a disease. And one of those factors is the infectious dose of the microorganism. Infectious dose basically refers to an estimate of how many cells it takes to cause a disease state. And there's a relationship between infectious dose and virulence, how nasty the pathogen is. So here's some estimated infectious doses of selected pathogens. It talks about the agent, the estimated infectious dose, and the primary route of infection. We'll talk about routes of infection in a little bit. So we've got measles, Q fever, plague, cholera, etc. Okay? And notice that they have very different infectious doses. Some of these are viral diseases, some of these are bacterial diseases. Did anybody hear in the news that they found someone in California just within the last week that has been infected with plague?
It's also a
Girl in Oregon? Okay. Yeah, the dreaded black plague, bubonic plague. It's still out there. Fortunately, not very common. Transmitted by fleas. Which is a more virulent disease? Cholera or measles? Measles is highly virulent. Why? Because it only really takes, theoretically, one measles virus. The bacteria that causes cholera, Rhinobacteria diphtheria, Vibrio cholera, is not a very virulent microorganism. It takes hundreds of thousands of cells in order to establish a particular disease state. Okay? Plague is much more virulent than cholera is. So just because you become associated or exposed to these particular microorganisms, if they're not very virulent, then they likely won't cause a disease state. That's why many of you are able to harbor microorganisms that are not highly virulent in your GI tract and not get sick. And you are then a carrier for that particular disease. However, if you then shed microorganisms through feces from your GI tract into water, and the water isn't properly treated, and somebody else that doesn't have as high a degree of immunity drinks that water, you can get cholera then from them. Does anybody know one of the most historically infamous carriers of disease? A woman by the name of Mary in the 1920s worked in the New York food industry, and she was a carrier for, anybody know? Is that terminology not still out there? Somebody who seems to make everybody sick, we used to call a typhoid Mary. Okay. Mary spread typhoid all around the city of New York through food handling. She was a carrier, an asymptomatic carrier. They actually imprisoned her and institutionalized her because she refused to stop working in the food industry. She would start working under assumed names and things and in infect new people. So it was a really very interesting story about somebody's civil rights being taken away because they were a carrier for a disease. Well, microorganisms have to be able to get into your body to cause infection. And typically, there is some fairly well-defined region where they gain entrance that we call a portal of entry. And the portal of entries are not, surprisingly, areas of the body where there's oftentimes the normal microbial flora anyway. So here are some common portals of entry. Broken skin, insect bites can both affect epithelial tissues. The mouth, the nose, the conjunctiva of the eye, the placenta, urethra, vagina, penis, nose, mouth. One of the things that we'll see is that particular pathogens are usually adapted to entrance at a particular portal of entry. The flu virus is really very specific for epithelial mucosa cells, like those that line the respiratory tract. You can take flu virus and rub it all over your skin, and you'll never get the flu. But even just inhaling a flu virus can cause influenza. One of the things that makes specificity important is that the pathogen's got to be able to attach to the portal of entry. And there can be specificity in the attachment that occurs. Certain types of cells have receptors, and they bind with various molecules on the surface of the pathogen called ligands. Ligand is just kind of a general term for a binding molecule. Sometimes these ligands are referred to as adhesins. So if a bacteria has a particular adhesin, it's got to match up with a receptor at a particular portal of entry. Some microorganisms can exploit multiple portals of entry. Tuberculosis can do that. There are two major routes of tuberculosis, and one is inhalation, but it also can be gotten by ingestion. Those are two very different portals of entry, and tuberculosis is pretty interesting in that it can exploit those two different types of entry points. Most bacteria are much more adapted to a particular portal of entry. So, if all this infection business begins at the portal of entry, the first step in that really involves attachment. You've got to get a foothold, because as we're going to see later, there are some defense systems that the body has to try to either prevent attachment or dislodge microorganisms that have become partially attached. So the first step in infection really is adhesion or attachment. And then if they can make their pathway into the cells, then we have this invasion. And the microorganisms start to multiply and grow or spread. If they become established in specific tissues and then cause damage to those tissues by these biochemical substances or virulence factors, then we can have a true disease state. Ultimately, there is typically a portal of exit too. And that gives the pathogen a way to get out of the host and then to infect new hosts. Those inept parasites, however, carry out this process so fast that many times they kill the host before they really have a chance to be shed. Okay? Portals of exit can include genitourinary tract or sexually transmitted diseases. They can be fecal in nature. And there are a number of different portals of exits that are involved as well. There was a concern for quite some time that the HIV virus was able to exit the body in sweat. And therefore, if you rubbed against someone that was sweaty that had HIV, you could become infected. That is not a portal of exit for HIV. So what we're going to do is we're going to start looking at some of these virulence factors, and we're going to break them down into three categories. So we'll kind of talk about each category and talk about some diseases associated with those particular virulence factors that we know a lot.